So welcome back again to the next presentation in sequence. If you've taken some time to think about these concept questions, here's what I hope you were thinking about. So what effect would the absence of oxygen have on the function of the electron transport chain? It would shut it down because the flow of electrons through the electron transport chain is dependent upon oxygen being available to accept those electrons at the end. If it's not present, then the electrons don't flow through because eventually all of the proteins in the electron transport chain are in their reduced form and therefore unable to accept any more electrons, and the flow stops. So we could say here, the ETC, that's the abbreviation for it, shuts down, and ATP synthesis stops. You die. Not a good outcome. The function of the electron transport chain relies on the fluidity of the Christie membrane. What feature is evidence for this? That would be the two mobile proteins that carry So while it's not really important that you know the names of the proteins, if you're looking for them on the diagram, one's called Q, the other's called cytochrome C. And you can see them in the diagram connecting the mobile protein, the complexes together. So it's these mobile proteins moving backwards and forwards inside the membrane that really reinforces the fact that the membrane is fluid, and that's what allows this the flow of electrons to continue in the electron transport chain. So moving on with the next section, we're going to be talking about what happens when oxygen is no longer available. Does that truly mean that everything really does stop and you die? Yes and no. It depends, as you'll see. So one of the big deals about cellular respiration, and specifically glycolysis, which remember is again the universal part of cellular respiration, the only part that occurs in the cytoplasm, the only part that occurs in the absence of oxygen, and the only part that every cell does if it does cell respiration at all. And the reason why I say that is because there are some organisms that don't have functional mitochondria, but they are all parasites, and they grab their energy from their host. So they don't really need to do cell respiration. But for any organism that has a dependence on glycolysis, there is a real issue, because it turns out that in cell respiration, in glycolysis, you can only make ATP after you do the reduction of NAD plus to NADH. It's only after that that the two steps that actually generate the ATP during glycolysis occur. So if you can't reduce NAD, if you don't have NAD plus to reduce to NADH, you can't make ATP in glycolysis. It's that simple. You can't run glycolysis without a constant supply of NAD+. Now, for cells that carry out full aerobic respiration, that's not a problem, because remember, the NADH that is formed by glycolysis, or as a product of glycolysis, transfers its electrons onto either NAD+ or FAD in the mitochondria, and therefore gets oxidized back to NAD+ again. So you have a constant supply of NAD+. But that relies on having functional mitochondria, which relies on, number one, having mitochondria, and number two, having access to oxygen. But what if you don't have functional mitochondria, or you don't have access to oxygen? How can you then reoxidize the NADH back to NAD+ to allow glycolysis to continue and to generate the ATP that you can get from it? Well, that's where fermentation comes into play. Fermentation provides the alternative pathway for oxidizing NADH back to NAD+. And there are two basic forms of oxidation of fermentation, what is referred to as alcoholic fermentation, which is carried out by yeasts, so yeasts are the one major group of eukaryotes that can actually be anaerobic or aerobic without any harm. They can flop backwards and forwards between the two. If there's oxygen present, they're fully aerobic. If there's no oxygen present, they revert to being anaerobic. And we take advantage of that, because when you look at alcoholic fermentation, it's a two-step process. So you take the pyruvate as the end product of glycolysis. In alcoholic fermentation, you do remove the carboxyl group. So there's the carboxyl group that gives rise to carbon dioxide. So this is sounding a whole lot like pyruvate oxidation. So this is actually a form of pyruvate oxidation. Instead of getting the acetyl group, however, we get acetaldehyde. And the acetaldehyde is then reduced to alcohol using the electrons from NADH, and that results in the NADH being oxidized back to NAD+ and allows glycolysis to keep working. So when we make alcoholic beverages, adult beverages, we're relying on the yeast to carry out fermentation and produce the alcohol, which means that if you are interested in making alcoholic beverages at home, because you can make some at home, it's allowed. You have to make sure that you exclude oxygen, because if you allow oxygen in, the yeast will become aerobic and you're not going to get any alcohol. All you're going to get is a sugar solution, and it's not going to be terribly pleasant. It's going to be very high in sugar and not much of anything else. But we also make use of this when we use yeast for baking bread and anything else where we use yeast as a leavening agent, because it's the carbon dioxide that actually causes the bread to rise, and anything else that we have yeast in, that is where we're using the yeast to make as a rising agent. It's the carbon dioxide that they're producing as they're anaerobic. They also do make a little alcohol, but when you bake the bread and anything else that you're using the yeast in, that alcohol gets baked out of it. But you can alter the flavor of your bread by using different types of yeast. So if you've ever tried sourdough bread actually relies on naturally occurring yeasts in the environment. That's what the sourdough is, the sourdough starter. It's actually utilizing yeast out of the environment. And you know that sourdough bread has a very distinctly different flavor from regular bread because it's using a different type of yeast. And the particular type of yeast in San Francisco apparently makes a superior form of sourdough bread. So San Francisco sourdough bread is famous as being the best type of sourdough bread. But you can make a sourdough bread anywhere using the natural yeast. There are yeasts in the atmosphere all around us as it is. In the wine growing regions around the world, it's the naturally occurring yeasts on the grapes that occur to the give rise to the different types of wines and so forth that are being produced in concert with the types of grapes that are being used. So regional yeasts make a big difference, as any wine producer can tell you. So that's alcoholic fermentation. In the case of lactic acid fermentation, this is bacteria and mussels. So your mussels are capable of carrying out lactic acid fermentation, but not terribly efficiently. It produces a really small amount of ATP, and that's the big problem. So in lactic acid fermentation, it's the pyruvate that is directly reduced to lactate. And of course, again, we get an oxidation up here. The NADH is oxidized to NAD+. That allows glycolysis to continue to make the small amount of ATP. For bacteria, this is not an issue. They have evolved to function like this. They can survive on the small amounts of ATP. And remember, again, a small amount of ATP is better than nothing. In the case of your mussels, no, this is not really good. They cannot continue to function on the small amount of ATP that's being produced by lactic acid fermentation. And if it continues for an extended period of time, your muscles complain and they stop working. You get a Charlie horse or a cramp. And that's the evidence that your muscles have gone seriously anaerobic and cannot produce enough ATP to keep functioning. And they literally stop functioning under the circumstances. And the only way to restore them to normal activity is to stop and completely aerate the muscle again, reoxygenate the muscle. It's the reason why after exercising hard, you continue to breathe hard, because you're trying to clear out all the lactic acid that has built up in the muscles by reoxygenating the muscle and processing that lactic acid through the mitochondria by oxidizing the lactic acid back to pyruvate, or the lactate to pyruvate, and then the pyruvate re-enters the aerobic pathway and gets processed through the mitochondria. Now, there is evidence that suggests that the lactate, or the buildup of lactate in the muscle, is what is responsible for the muscle cramp. But there is an even larger body of evidence, more recently obtained, that suggests that the lactate does build up, but it does not stop the muscle from cramping, because you can set up a situation where you can get the lactate out of the muscle experimentally and still find the muscle cramping, even in the absence of lactate building up in the muscle. And so we're not entirely sure what causes a muscle cramp, although it's probably got some relationship to the low levels of ATP, which are necessary for the muscles to work. But it does not appear to be directly correlated to the lactate. The lactate may be a symptom that leads to the muscle cramp, or that the muscle cramp is coming, but it may not be the direct cause of the muscle cramp. But it may be a symptom of what is going to give rise to the muscle cramp. There are a number of other ways of doing fermentation. And so you can see some of the other materials that are produced by different types of bacteria. And so Propionibacteria, Clostridium, E. coli and Salmonella, Enterobacteria, these are all not particularly pleasant bacteria, particularly these three. These are responsible for food poisoning. E. coli lives in our in our colon. It's supposed to live in our colon, but when it gets into other parts of our digestive tract, not a good thing. Same with salmonella. Clostridium tends to grow in poorly sterilized canned food. So if you find a can that's swollen on the ends, don't even bother opening it. It's just telling you that something inside desperately wants to get out. Never a good thing. And so Clostridium, both botulinum, Clostridium botulinum and Clostridium tetani are really bad news. Botulinum is what causes botulism. Ironically, it is the toxic substance that is produced as a consequence of the presence of these bacteria that also leads to food poisoning, is what is used in a very dilute solution in Botox injections. Why? Because in both situations, it paralyzes the muscle. In one case, it paralyzes the muscle and kills you. In the other, if it's done correctly, it paralyzes the muscle and smooths out the wrinkles, but done incorrectly, it will kill you too. And people have died as a consequence of poorly given Botox injections. Tetany is the bacterium that gives rise to tetanus, and again, it causes muscle spasming. The old name for tetanus is lockjaw, because it used to cause the muscle to spasm and lock the jaw tight, and you couldn't even force it open because of the presence of it, and it would wind up killing you. Enterobacteria, not quite so nasty, but nasty, and the same with Propionibacterium. Propionibacterium, by the way, one type of Propionibacterium is what is responsible for zits, acne. They grow in blocked pores on your skin, and they grow anaerobically in the pores in your skin. They also lead to the death of white blood cells that are trying to get rid of them, and that's actually what causes the zit, the white material. That's actually dead white blood cells. All sorts of interesting things that we can discover if we dig a little bit deeper into this. Anyway, you can see that there are a number of different ways of doing fermentation other than lactic acid and ethanolic, but these are the major pathways. So when we start thinking in terms of cellular respiration and fermentation, and we take a look at the key point in both pathways, well, the key point is pyruvate. If there is no oxygen present or no functional mitochondria, then fermentation is going to take place. One of the multitude of forms, primarily ethanolic or lactate fermentation, but as I showed you, other types. If there is oxygen present and functional mitochondria, then the pyruvate enters the mitochondria and get processes through the complete aerobic pathway. Now, there is also a type of respiration called anaerobic respiration. Sometimes that's used to refer to fermentation, because anaerobic literally means in the absence of oxygen, and that is a good way to describe what's going on where fermentation is involved. But it's also applied to circumstances where, rather than using as oxygen as the electron acceptor at the end of the electron transport pathway, you use another molecule. So one that you might be familiar with the product of is one that uses sulfur dioxide as the acceptor molecule of the electrons. That produces another molecule called H2S, hydrogen sulfide, otherwise known as rotten egg gas. And that is also known as swamp gas. It's if you've got a bad drain at home that's dried out, you may smell that because it's also known as sewer gas. So sewer gas, swamp gas, rotten egg smell, or rotten egg gas, it's all the same thing. It's H2S, and it's produced by anaerobic bacteria that are using sulfur dioxide as their electron acceptor, not oxygen, to allow them to run their electron transport pathway. And there are a number of other molecules that can be used as electron acceptors as well to allow for non-oxygen cell respiration, a form of anaerobic respiration that uses the full cell respiration pathway but doesn't use oxygen as the receptor molecule for the electrons. And glycolysis, of course, as a non-oxygen requiring part of cell respiration, gives us some clues as to where cell respiration may have come from. We know that oxygen was not a major constituent of the atmosphere until oxygenic photosynthesis, what plants do and algae do, became a major chemical pathway. We'll talk a little bit more about that in the next chapter. But that means that making ATP could go ahead in the absence of oxygen so long as the glycolysis pathway was present. So glycolysis is probably a very, very ancient pathway. It is reflected in the fact that everybody does it, both prokaryotes and eukaryotes, and everybody does it in the absence of oxygen, which is a reflection of where it probably evolved. So it gives us some clues as to where some cellular biochemistry may have come from. So that's fermentation and anaerobic respiration. So it's time to take another quick break, and I am going to stop this recording at this point and pick it up again with one more recording that's going to cover the regulation of cell respiration and its biochemical flexibility, as you'll see. So here's the concept check question for you to think about while you're getting ready to watch the final animation. Compare and contrast aerobic and anaerobic respiration. Include a brief description of the processes involved. I'll give you the answer at the next recording.